So here are Koch's postulates. There were four of them. One, the microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. We're going to come back to that in just a second. The microorganism must be isolated from a diseased organism and grown in pure culture. So in other words, if there's somebody sick, I should be able to take whatever's making them sick and grow it separately on a culture plate. 3, the cultured microorganism should cause disease when introduced into a healthy organism. So the guy that discovered H. pylori caused ulcers basically did this. He isolated H. pylori from people with ulcers. He was able to grow it independently, and then he gave himself and his wife H. pylori, and they got ulcers. So once this is done and that person or animal or whatever is sick, you should again be able to re-isolate whatever's making them sick. That sample should be the exact same match to this sample that you took from the original person that you got it from. Now, let's talk about one of the things that he changed. So these were his original postulates, that's true. But this first one, it should not be found in healthy individuals. Technically, you can have carriers that aren't exhibiting symptoms of the disease. Typhoid Mary was a real good example of that. She was a nurse and she had typhus, and she went and worked in hospitals and was spreading typhus. She never exhibited the symptoms of the disease, yet she was still able to spread it. So when he found out that there were carriers, he actually changed that first postulate. So recent advances in micro. The discovery of restriction enzymes in the 1970s. Basically, restriction enzymes are enzymes, things that cut proteins, or cut things, break them apart, that allows you to cut at specific points in on DNA. Okay, so it allows you to kind of pick the sequence that you are going to cut at. This has actually really opened the doors for us as far as genetic engineering, because it has allowed us to cut at a specific point in a plasmid, a little ring of DNA, and then cut what we're interested in at those same points so that we can insert it and it fits together properly. Now, the invention of polymerase chain reaction, PCR. This happened in the 80s. It is a DNA amplification tool. If I have DNA, I can use PCR to make multiple copies of that strand of DNA. It helps us to identify new organisms. It also helps in forensic work. So when, let's say, a woman is attacked and she scratches her attacker, they can take the samples from under her nails and actually make more DNA than what would originally be just under her nails. It's a way to get more so that you can look at it with a lot more detail. The importance of biofilms. This is from the 80s on. They're now recognized as very common on surfaces and key in causing certain infections. So biofilms are a collection of bacteria, almost like a community of bacteria that stick to, for the most part, when we talk about them, medical equipment. And the thing is, the way that they stick, the way that they make this community of bacteria, it actually makes it really hard to get rid of them, because it isn't just one bacteria, it's a lot of them. And they kind of have layer after layer, and the outer layers protect the inner layers. So when we get to talking about biofilms in a lot more detail, we'll talk about the fact that in order for that not to happen, especially in hospitals, the equipment has to be cleaned in a really specific way to prevent that biofilm from staying. a while back, there was a rash of infections at different hospitals all over the country. And what they figured out was that they were all using the same company to clean all of their equipment. Well, the company wasn't doing it correctly. So biofilms were forming in all of this stuff and basically being transferred to the patients at the different hospitals. So again, sorry, autoclave. Cellular organization of microbes. Eukaryotic microbes. Okay, like I said, eu means true, karyo means nut, it's a nucleus. Eukaryotic microbes are more complex single cell organisms that actually have organelles, specific parts inside the cell that have specific jobs. Okay? So you have organelles that are membrane bound structures that perform specific functions inside the cell. Fungi are eukaryotic. Protozoans are eukaryotic. Helminths are eukaryotic. Prokaryotes, remember pro is before, karyo is nut. These are the simpler cells. They do not have organelles or a nucleus, which is an organelle. They're actually about 10 times smaller than the eukaryotes. They lack organelles, like I said. Bacteria, just simple bacteria, are going to be prokaryotes. And archaea, which are what we call the extremophiles. You find them in volcanoes, you find them in icebergs, you find them in those types of places. Um, they're the simple cells. Now, viruses are a bit different. Um, Bacteria can live by themselves for the most part. Eukaryotes can live by themselves. So these two can live on their own in the right conditions. Viruses can't. They are not independently living cellular organisms. They're composed basically of genetic information, whether that's DNA or RNA, in a packet, a protein coat packet. Okay? They don't have a nucleus, they don't have any organelles, they don't have a cell membrane, they don't have any of that. They are basically going to be parasites to your cells. So when you get a virus, that virus goes into one of your cells, takes over the machinery of that cell, and makes it into a virus factory. The thing is, the virus by itself can't do that at all. Literally, it doesn't have the equipment, it doesn't have anything other than here's my genetic code in my DNA or RNA, and I'm encased in a capsid or a packet. So it is a type of microbe because it is microscopic and it can cause infections. So it's pathogenic. But ultimately, it isn't independently living. I hope that makes sense. So, naming and classifying microbes. The way that we classify everything, just general umbrella, is called taxonomy. It's the science of classifying living things. So it could be an elephant, it could be a whale, it could be Staphylococcus aureus, a bacteria. Binomial system of nomenclature. Bi means two, nomial means name. So it's a method of assigning a specific name, a scientific name, to an organism. And it is composed of two names. Okay? The first name and the last name, just like I'm Ronica Williams, these actually have first names and last names. So the first name is the genus name, and it is always capitalized. Okay? The second name is the species name, and it's always lowercase. Give me 1 second. So for humans, our scientific name is Homo sapien. Okay, notice the capital letter here, the lowercase letter here. This is the genus name, this is the species name. Now, when you handwrite these, when you put them into papers, you basically have two options. Handwriting, you underline it because it's a proper name, it's a taxonomic classification. If you're typing it into a paper, you would put it in italics or you can underline it. Unfortunately, with writing, you can't really do italics. But for it to be properly represented, it either needs to be in italics or it needs to be underlined. Okay? So again, Homo sapien is our scientific name. When I mentioned Staphylococcus aureus, that's the scientific name. So Staphylococcus would be capitalized, aureus would be lowercase because it's the scientific name. It's the taxonomic name. One second to erase this. Okay, so when we classify organisms, you kind of have the huge umbrella that kind of covers everybody, and then you kind of start getting more and more specific. So, for example, the biggest umbrellas are the domains, okay? Then you have the kingdoms, smaller umbrella, the phylum, smaller umbrella, the class, smaller umbrella, order, family, genus, species, until that's the smallest umbrella you got. So, um, okay, to remember the order, did King Philip come over for good spaghetti? Okay, that'll kind of give you the order and gives you the first letter. So hopefully that'll kind of give you an idea of who's next in the series. Okay, so there are three domains. Remember, domain is the biggest umbrellas. Okay, so there are three domains that contain all life forms: archaea, prokaryotic cells that live in extreme environments. Like I said, these are also known as the extremophiles. They are in volcano vents, they're in icebergs, they're in the hot springs at Yellowstone National Park. These are not things that are found on a table because the minute you take them out of that extreme environment that they live in, they die. Okay? Then you've got the bacteria. These are prokaryotic cells that include most of the diseases, of the disease causing bacteria. Eukarya. Eukarya is all eukaryotic organisms. It includes algae, protozoans, fungi, plants, and animals. So trees, rose bushes, us, bears, lemurs, hippopotami. It includes all of that. So you've got this kind of classification system that, like I said, you go from big umbrella to smaller to smaller to smaller to basically a focus group. Okay, so that's chapter 1.